Do, do, do. Good morning, everybody. Morning, Tanya. Oh, just beat your pipe, Rose. Okay. Bit pie-eyed, feeling like a need for a spring break, unfortunately, which we're not going to have. So, I'm not quite as bright-eyed and bushy-tailed as I should be this morning. Okay, so you're going to have to do me a favor and remind me where we finished up last class. Hang on. Can you all hear me? Anybody? You can? Oh, cool. That's good. Sorry, Rose, that's just you. Don't know why. Perhaps turn your speakers on? Sometimes, Rose, if you sign out and sign back in again, it works again. Zoom can be kind of funky depending on what else has been running. I know WebEx completely screws things up from experience. Okay, so where did we finish? Did we do the 2-point cross? Because I can't see the YouTube thing because my computer's refusing to play back videos for some reason at the moment.
So the last thing we were doing was the RF recombination frequency.
Okay, let's see. Do. Did we do this?
Yeah.
We did. Okay, I'm sorry because I don't have any pictures from that class, so I'm kind of struggling to figure out where we were at. Did we do this? No.
No.
Did we do this?
Nope.
Awesome. Okay, so do this then. Ha! There you go. You can make yourself a cup of coffee. Oh, that was just a joke. Although I would love. I wonder if I can convince my wife to make me a cup of coffee. Okay, so what I want you to do is figure out a plausible map from these data. Right, so remember, anytime we have less than 50% recombinance, these genes are linked, right? So this is an extreme case of linkage here. So I want you to figure out what order these genes are in relative to each other. Give you a few moments to do that, and then we'll work through this problem. And remember as well, anywhere you have 50% recombination between them are so far apart they are linked. However, you can't tell how much further apart they are. It's like if your speedo only goes to 70 miles an hour, you can't tell once you go over that speed how much faster you're going than 70, only that you're doing at least 70. While you're doing that, I think I'm going to go get some puppies to show you. Okay. You can get one each, girls. All right. This is, yeah, I can take it. This is Nikita, or Nikki for short. She's super cute. She's got really, really short legs and fluffy ears. And a big belly too. See?
She looks mildly displeased.
Hang on. And this is Lily. Yeah, she's got a sweater on because she's a little bit sick. Anyway, anybody want a dog? She's lovely. There you go. There you are. All right. So, a little bit of puppy interlude. Okay, so let's figure out a map, right? Because we've got all of 3-point crosses to go to as well. Okay, first off, which is the furthest gene from all the others?
A.
A. Right. I'm going to do this from left to right just because English is my language, I guess. And we write from left to right. Let's actually turn this around. So we have line representing our chromosome. We have A over here. And over on the far side, we have B, C, D. Actually, I'm just going to do this a little bit higher, give myself a bit more space. Right. And we know that A is all the way out here on its own because of what? Why is it not? Why do we know? How do we know that all of those three are all together and far away?
The recombination frequency is high.
It's at 50%, right, for all of them. So it's, let me just turn this up a bit. So we have 50 centimorgans between these. Now, we don't know which way round these are. We don't know if which is closest out of B, C, or D to A, because they are all unlinked from A. Right, there's no linkage whatsoever, or there's no linkage between A and any of the other genes. Okay, so what we need to do now is figure out the relationship between B, C, and D. Okay, so which is the closest pair to each other?
B and D.
B and D. Whoever that is, can you speak up just a little bit when you ask? You're completely correct. I just not hearing you very well. I've got giggling girls outside. Okay, so we have A and we have B and D at 10. Right, now, is the order this, or is it that? How can you tell?
It's CBD.
CBD, why?
CD is it's larger with 30.
CD, did you say?
Yeah, CD is farther away than B and C.
right? And also, so one way you can do this is trying to figure out what doesn't work. So this is 10. If we had BC is what, 20? Then D to C would have to be 10 as well, or C to D, and it's not. Right, so one way you can do it is by adding things together, okay? So C and D is the sum of B and C and B and D, right? 20 plus 30. The other way is you can do it kind of subtractively as well, right? Which one of these fits, right? So this is 30. Make sure you can see that. So this has to be 20. And that works for all of them, right? So this one, as several of you already said, is wrong, right? Now, do we know that it's going to be A, C, B, D? Is that the correct order?
We have no way of knowing.
Yeah, who is that, by the way?
This is Gabe.
Say again?
Gabe.
Oh, I can't hear you, sorry. I just can't see who's talking. I'm just nice to know, that's all. So you are 100% correct. It could also be D, B, C, right? And that would be consistent as well with the distances between the genes and the distance between A and this group as well. So the only way that you could figure out which of these overall orders are correct, A, C, B, D, or A, D, B, C, is by finding something in the middle that is linked to both A and to at least one of these. And then you can work out the order of the whole thing. Right, so you quite often it depends on how far apart your markers are as to how complete your map can be. Let's just take a picture of that before I forget. Okay, any questions about that before we move on? Nope. It's relatively straightforward. A lot of this is really just math at the end of the day, and it's fairly straightforward math as well. The only thing you have to remember are the rules. Okay, so 2-point crosses are all well and good, but as you can see, you have to do an awful lot of them to be able to figure out gene order. And sometimes you can't actually figure out gene order very well, right? The other issue with 2-point crosses is that you don't pick up, double crossovers. So you can get 2 crossover events on the same chromosome arm. It's rare, but they can occur. And if you're only looking at A and B, right, separately, and you don't have a third one in between, you will not detect those double crossovers, and those still count as crossovers, right? And so that's going to give you a inaccurate measure of distance, right? Because you're not accounting for all of the recombination that's occurring between them. And so the way of dealing with that and also getting gene order is to do a 3-point cross. And so essentially a 3-point cross would be looking at B, C, and D in one cross, right? Rather than doing 3 separate crosses to get that information, right? So it's a lot quicker for a start, and it's also more accurate, which is kind of a win all round. So essentially, and comfy. When you're doing a 3-point cross, right? First, you need to have 3 different loci, right? A, B, and C in this case, right? And the same deal applies with, as with a 2-point cross, you need to have an individual that is heterozygous for all of those, right? So notation is kind of all over the place, unfortunately, right? This would be plus, 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 and this would be A, B, C. These are using capital letters instead, right? But when I'm working at stuff up on the whiteboard, I'm going to use plus to denote dominant, right? Just to kind of try and keep things a little more consistent. And so same deal applies. You're going to have a heterozygous parent. You're not going to know, as before, whether these are in cis or in trans. Each one of these pairs, right? It could be plus BC, or it could be plus plus C, or it could be plus plus plus, right? You just don't know until you look at the cross progeny. Also, you cannot know the gene order, right, before doing the cross. So you may be given a gene order, right? And that's kind of the, well, we're just going to give you the genotype, right, of the parents. But you have to work out from the numbers what the actual gene order is, right? This is really, really important. Do not assume that the order of letters that you're given is the correct one until you work out whether or not that is correct. It may be simply putting the genes in alphabetical order, or just write them down in whatever order that the person happens to write them, right? Only by actually doing the 3-point cross can you work out the actual gene order, right? Can't stress that enough. You can't assume anything going into one of these. You have to work it all out from the numbers, all right? And that's not to trick you. I don't want to be like kind of sneaky or anything. That's simply because the numbers are what tell you that information. Doesn't matter what you assume ahead of time, the numbers are what are the data, right? What are correct, the facts. Okay, so and let me say that for a third time. You can't assume the gene order, all right? Really important. You have to work that out. So maybe I should write that down in the chat box as well. When you do a 3-point cross, you're going to be having a heterozygote for all of those 3 loci, and you're going to cross that with a homozygous recessive for all 3, right? Because that's what you need to be able to see recombination. Very much the same as what we're doing in lab with the fly cross, right? With the linkage experiment there. When you look at the outcome of that cross, you will have 4 different categories, right? Each one of those will have 2 sets of organisms, because every cross will generate a reciprocal, right? So if you have a crossover between A and B, you'll have one offspring which is plus and B, and the other one has to be A and plus, right? They have to be the opposite mirror image of each other. That's just how it works, right? So of those 4 categories, you're going to have some pretty obvious differences, right? So given that we're expecting linkage to be occurring, right, what group will have the highest number of progeny? So one group, 2 different phenotype combinations. Which group will have the highest number of progeny if linkage is occurring? Think about it. The key is if linkage is occurring. Now, not so much talking about the genes here. We're talking about parental versus recombinant. Yeah, I just told you. Which group's going to have the highest number? Remember, linkage is always the characteristic of linkage.
Parental, right?
Correct. There's always an excess of parentals, right? Or a less than expected number of recombinants. It's kind of the flip of that, the other side of the coin. So one group, you'll have lots of wild type or dominant for all three in this case. And the mirror image of that would be lots of recessive for all three, right? So it'd be a homozygous dominant, homozygous recessive in this case, right? So those are the parentals. Can't assume that that's going to be the case, but whatever group is the largest, and there will be mirror images of each other, will be the parents. Right, so the largest group will tell you what the genotype of the parents is. Right, that's super important because you're going to use that to figure out where recombination is occurring to build your map. And again, these don't necessarily have to be all in cis, right? To be honest, most time, I think by and large, I'll give you it all in cis because you've got enough on your plate to work out without having an extra complication of the trans linkage, right? But you can't assume that. You work that out from the parents, all right? Then you're going to have a group that is a really small number, right? Those will be the double crossovers, right? And the reason for that really small number, or the smallest number, right, of all of those four groups, is because essentially these two crossovers are independent events. And so what is the probability? How do you calculate the probability of getting two independent events? Remember back to Mendelian inheritance? Its rule begins with M.
Multiplication.
Yeah. So you get the probability of this, a crossover occurring here, and essentially times that with the probability of recombination occurring between the other group. And that will give you the probability of getting both. So just mathematically, you would expect double crossovers to be the rarest, right? And the other two groups are going to be single crossovers or the result of single crossover events between these two genes or the other two genes, between A and B or B and C. These two groups do not have to be about the same. A and B could be far, far closer to each other than B and C, or vice versa. Or they could be approximately the same distance apart. You've got no way of knowing until you do the experiment.
Okay, Dr. Crick, because this was kind of a lot for me. So there's going to be four groups. One is going to be like linkage because they're going to have a lot of parentals. The other two.
No, those are going to be showing no recombination, right? Essentially,
Okay, no recombination because they're going to be like the parents, right?
That's right, because these are going to be, there's going to be two possible reasons for getting a parental chromosome, right? Remember, one reason is that you're going to have parental chromosomes that never undergo recombination. They're on the outside, right? So at least 50% of the progeny of this cross will always be parents, always, right?
Okay.
Now, if you don't have recombination occurring, then you get only parents out of that, right? So if linkage is occurring, which we're assuming it is, between these two genes, these three genes rather, then at some point you're going to also get not just the parents on the outside that never recombine, you're also going to get the non-sister chromatids which can recombine but don't, right?
Okay.
Because linkage is occurring, because there is a lower than expected rate of recombination in the things you're looking at.
Okay, so the first group are going to be the parental that they non-recombinant. And then the second and third are going to be the recombinants. And they're
Single recombinants.
okay. And then the last one is going to be the smallest because that's where they double combine.
Correct.
Okay, got it.
Yeah. So this is the purpose of going through it in this way, is really to, I don't know, introduce you to this idea slowly, right? Because if I just show you a bunch of data, it can be really overwhelming, right? And one thing I can guarantee you is that once you really get how to do this, you'll be like, it can't be that easy, right? Promise you that. I've had students just yesterday go, wait, there's got to be some kind of trick going on here. I was like, no, it's really just it's that straightforward, right? But until you get that, it's going to look like a complete horror show, right? Okay, so one really, really important thing to do. Okay, so here's my stepwise plan. This is Dr. Matt's patented 3-point crossover plan for success, right? First, figure out the parents, right? Let's type this down. Figure out the parents. And those have the highest number of progeny. Okay, that's easy, right? You're just going to look at the data you get. They're not necessarily going to be in order, right? They might even be all jumbled around. You just have to look for the biggest numbers, right? 2, right? Oh, and that will tell you the genotype of the parents. So e.g. ABC, or I don't know, it could be plus BC, A++, or whatever, right? Two, find the double recombinants. That would be the smallest number of progeny. Progeny. Ah, man, I can't spell this morning. And that will tell you the gene order. Remember, what you get given to begin with may not be the correct gene order. And the reason why the double recombinants will tell you the gene order is because the one that moves is in the middle. Okay, so the only way you could get a plus B plus and an A plus C recombinant is if B is in the middle. If
Can you say
you're
that again?
the only way that you can get, for example, if The only way that you could get B moving alone, right? So if you look at the, oh, crabs. Come on, let me get back to, there we go. Yes, that's a lovely bone, Luna. My dog showing me a bone. Right, so the parent is plus, 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 or capital A, capital B, capital C, right? Or ABC recessive. So in your recombinants, you're seeing you've got dominant and dominant for A and C, and recessive for B. The only way that you could get that combination of alleles, dominant A, recessive B, dominant C, is if B is in the middle. Because essentially you're cutting out the bit in the middle and sticking it in the other chromosome and vice versa. It's not the end of that sequence, it's the one in the middle. Right, so for example, if you got instead, it would be recessive A, capital B, capital C, and that was the double recombinant. And the flip side of that would be dominant A, recessive B, recessive C. The only way you could get that would be if A was in the middle, because A is the only one that's moving. And you know that these are double recombinants because they have the lowest number. Right? So for the lowest number of progeny, whichever allele moves, and this is why you need to know the genotype of the parents, right? Because that's the only way you can figure out which allele is moving, right? Once you figure that out, then the one that's moving is the one that's in the middle. That's what gives you your gene order. Does that make sense? Again, we're actually going to go through a bunch of examples. So this is kind of like the theory of 3-point crosses right now. And you know, like theory is a lot harder to get than actual applied practical stuff.
Because I was reading the book and I got lost and I was like, I'm just going to wait for a lecture because they explain it, but it's different to actually like visualize it and then
Right.
making sense of it.
Well, that's why I have Dr. Matt's patented 3-point plan for success, right?
Yes, it's making sense. The steps are going to follow for another one. It's making sense.
Good. And like I said, once you get this, it's like, wow, that's easy, right? Promise. You're not going to believe me right now, I know, but trust me, I'm a doctor. Anyway, third step in the plan for success. Work out recombination frequency for each pair in turn. Now, there is a little snag here, right, which you cannot forget, right? So RF for, let's say, A, B equals number of essentially A, B, how would I write this? Right, so this is the number of single recombinants. Okay, that's pretty obvious, right? Essentially, it's going to be this group here. But I'm going to move that over here. If you have double crossovers, you are also still getting recombination between A and B. I mean, you are getting recombination between B and C, but we don't care about that for calculating the recombination frequency between A and B. But we do care about this one, right? So to get an accurate recombination frequency and distance, you need to also include the double crossovers, right? That's for AB, and then you do the same for BC. And that's it. Nothing more to it than that. Now, give you time to let that sink in. And then we'll do a bunch. I've actually worked one up for you, which you don't have here, which is kind of neat. Very proud of myself. So just to reiterate, first, what are the parents? That gives you the genotype. Second, what are the double recombinants? That gives you the gene order. Third, work out recombination frequency by adding single and double recombinants for one pair, A and B or B and C, let's say, right? That's it. All right, you're ready to move on? Any questions? Okay, cool. So this is what one of those looks like. You should have 8 combinations. Yep. You're going to have, essentially, you can have either phenotypes or you can have the genotypes of the gametes, right? Essentially the chromosome that you care about, which is from the mother. This is telling you the genotypes of the parents. And again, this is not the gene order, right? This is just the genotype of the parents. Sometimes you might not even be given that, right? But you can always assume that you're going to have a parent that is heterozygous for each of those loci, for each of those genes, crossed with a homozygous recessive, right? Or if it's X-linked, then a recessive male, right? Just the same as you're doing in the lab. Okay, so first step, which group have the most progeny?
The first one and the 8th one.
Correct. So wild type, so that's going to be dominant, 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 and recessive for all 3, which is going to be recessive, 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 right? So that just confirms what you've already been told up here, right? So this is the correct genotype for the parents. Doesn't hurt to just double check. Right, so these are the parents, the largest numbers. That's all we're going to be doing with those from now on. Those are not part of any of the calculations. Right. Step 2. Which ones are the double recombinants?
The sugary and the lacy glossy.
Right. I don't know if those are actual real or not, but they might just be made up. Doesn't really matter. Okay, so you can see very clearly, right? It's not always going to be as clear as this, but these are definitely the ones with the smallest number. Okay, so let's actually work this up on the board. Right, because this is, I'm not going to try to describe this stuff in words. Right, 3-point cross, number 1. Okay, so we have, I'm just looking at the stuff. These are our, I hate using capitals. It's so clunky. Right. So these are our 2 parental chromosomes. Yep. And our double recombinants, see, these are parents. Our double recombinants are this. Which allele is moving? That's 100% correct. This one here is the one that's moving. So is this gene order correct?
No, should it be in the middle?
Correct. Oh no, it is wrong. You are correct, right? So the correct order is LZ, SU, GL. Because that one is the one that is moving in the double recombinants. Let's see if I move this back a smidge. Take a picture of this. That doesn't include my knee. There we go. Does that make sense? So I'm personally a big fan of the whole turtle approach to logic puzzles, right? I think it's, if you take it slow and you follow the steps, you can't get it wrong. It's like doing a chi-squared test or any other, or dihybrid cross or any of this stuff, right? As long as you don't jump to assumptions and try and race ahead and just do things in a nice you know, turtle wandering along the road kind of way, you'll always get it right. Okay, so I'm going to cross this out and I'm going to put this correct gene order up here. Okay, because then we're going to use that to figure out what the single recombinants are. Because that will make life a little easier. Okay, so this is the correct gene order. So now what we need to do is figure out, okay, what are the progeny that result from a crossover here, right? So we need to look for the single recombinants. So we're going to have a dominant LZ, right? And then we're going to have these two from over here. You see why it just irritates the monkeys out of me having to do this with like letters and stuff. And likewise, we're going to have the recessive one of these, and then these two, right? So these are going to be, what was that, sugary and glossy? Going to be phenotypes, and this one's going to be lazy. Okay, so what are the numbers for those? So this should be just lazy, and this should be sugary and glossy, or glossy and sugary.
So 40 and 33.
Okay, 40, 33. Now, what else do we need to add? Remember, step 3.
Wait, I think it's the other way around. The lazy's 33, I think.
This one is lazy because this is the recessive. So this should be sugary because that's the recessive phenotype. This should be glossy. That's recessive. This one should be normal for sugary and glossy, so you don't see it, and then lazy. Okay. Right, what else do we need to add to figure out recombination frequency?
The sugary and lacy glossy
Okay.
of the double recombinants.
So the double recombinants are going to be just sugary and then lazy glossy. And this is why it's just such a pain in the butt trying to say this stuff. Right. Anyway, so then you also need the double Rex, which I'm not going to write up their genotypes, but we have 4 and 2. Okay, so recombination frequency. Oh, my daughter's hair stuck to that. Right, let's pull this back a little bit further. Okay, is 40 plus 33 plus 4 plus 2. What's the total? Divided by 740 times 100 to get us into the percent.
10.68.
0.68?
10.
Oh.
10.68.
10.68 centimorgans. Can you see that? There's a Matthew Crook right over the 10.68 when I have my computer like this. There we go. Everybody understand how we got that figure? Shout out if you don't. Oh, it's a hard life, dog. Okay. So now we're going to do I'm a little lazy to write that all out again. Right, so now if we want to work out the distance between SU and GL, now we need to find which ones are going to be glossy. And then the opposite, right, is going to be lazy, sugary, and that's it. Right, so glossy, there it is. right? How many glossy whatevers do we have?
59.
And the reciprocal, the opposite of that?
44.
Correct. It's going to be lazy and sugary. So you just plug those numbers in. And what do you get?
14.73.
So that's the distance between those two. So then all you have to do, once I take a picture and rub this off, is draw a map. You're going to have to remind me of what the distances are in a second. So your map would be this is 14.7, I think. Got
Yes.
terrible memory. And this is 10.68, I think, or 10.7. I think. Can't remember. And that's all there is to it. So what we're going to do now is actually, you know what I want to do first, just to make sure I'm not worried about time at the end. Just want to go over some stuff at the end of the PowerPoint, just to make sure that, you know, we cover it. And then we're going to do a couple of practices. Oh yeah, there you go. That's what it looks like. So you got all the answers there for you. Okay, and the reason why I want to do this first, even though this is technically last, is because then I'm just not going to worry about having to fit this at the end, right? So we can just keep on going until the end of the class. Okay, so essentially there are two different types of maps. One is a linkage map, right, which is based on recombination frequency, right? And so that's essentially what we've been working out. The second is a physical map, right? And that is the actual kind of number of bases, right? So for example, this is from Flybase. You'll be going here for part of your lab assignment, right? This is the linkage group for miniature wings, M. This is its physical distance, its physical location on the X chromosome, right? 11,754,233 bases to the next one, right? If you look at, come on, yellow, right, which is a gene we're working on in lab, right? Same chromosome, they call it 1 for some reason, I don't know why, it's the X chromosome. Now you can see that this is linkage group 0, right? So that's a long, long way away, right? And you can see that its physical location is also different, right? This is now in the hundreds of thousands, not the tens of millions, right, of bases, right? So that's really just the difference between those two. And typically speaking, there's a positive correlation between the two. The only thing to be aware of is that that breaks down for regions of low recombination, right, which are around the centromere. And so this is largely heterochromatin, right, which is really kind of like scrunched up, tightly wound DNA. And typically, there's much less recombination in this area, right? So physically, right, this may look larger than it does in terms of the genetic map because there's less recombination going on in here, right? So that's going to make it look like those 2 points on either side are a lot closer than they actually are physically, right? So that's just something to be aware of, right? Linkage maps are not physical maps. They're determined by recombination frequency, right? And so that's based on the assumption that recombination frequency is equally, the probability of recombination is equally likely at any point between those two different things you're looking at, which may or may not be the case. Typically it is. On the arms, it definitely is. All right. Okay. So let's do one that I worked up. So if you looked earlier in the PowerPoint, pretty sure we actually used these to do the single point cross examples. Right, we're going to be using white-eyed, diminutive males, and miniature wings. Okay, so let me write this down. So it's going to be, and I'm going to use the plus letter notation here just because it's a whole lot easier. Right, so you're going to have this individual right is going to be crossed with, and this is X-linked because it's on the X chromosome, right? So it's going to be a recessive male. So this is going to be a white-eyed, small-winged, small male with serious personality issues, probably. So you're going to get, and what I'm going to do is I'm going to write down the different combinations. I'm not going to write down the phenotypes. I'm just, well, I mean, essentially it's the phenotypes. I'm going to write down the genotype of the chromosome that you get from this parent, right? So this will be the result or not, as case may be, of recombination in this parent, right? So, and I'm going to make life easy because I'm just going to give it to you in group order, because otherwise my brain would hurt a whole bunch. And I'll make sure that this is all easily visible in a second. We have plus DM and plus, which are 3 and 2. And the total adds up because I like nice round numbers. 2. Yeah, you can just about see all of that. There you go. Give you time to write all that down. I've done you some of the hard work, or well, not hard work, but like slog work, I guess, by grouping these into reciprocal groups. We won't always have that but you know, I'm a kind of generous soul, so I figured I'd do that for you. So these are the groups. And you can figure that out too, because, you know, plus W, M plus, DM plus, right? So this is the reciprocal of this. Has everybody had the chance to write that down? Okay, so step 1 of my plan for success is what?
Parents with the highest number of progeny.
Okay, it's pretty obvious which ones those are.
The first two.
Okay, so these ones here are the parental chromosomes. What's step 2?
The double recombinant.
Okay, which one's those?
The bottom two.
Okay, so Tanya, let someone else give a chance to answer. What is the correct gene order? If you look at those, so this is step 1. This is step 2. What is moving in these 2 chromosomes compared to the original parental chromosomes? Which gene is moving? Which allele is moving? Yeah, good stuff, Gabriel. Okay, so DM is moving. What is the gene order? What is the only scenario in which a DM will move in a double crossover? What's the only gene order? Well, actually, technically there's 2, because it could be either way around. Yeah, thank you, Levia. That's exactly what it is. Right, so our gene order is actually wrong. Right, our assumption was incorrect. Get my knee out of the way. So the actual gene order is tangled up with my headphone. Right, that's the correct gene order, which we get from the double recombinant. Now, all you need to do is work out step 3. Is RF for W and DM is going to be any that result in crossovers here. So which of these is going to be, essentially that's going to be a W++ and then a plus DMM. So where's a W++? Going to be these two, right? So we need W++, which is 4. We need plus MDM, which is 6. And what other numbers do we need? What's the snag that you cannot forget? Recombination frequency is the number of single recombinants plus the number of double recombinants, exactly, which are these ones down here. Tanya, you can speak up if you want. I just wanted someone else to have a shot at the gene order, that's all. So all of that divided by 1,000 times by 100 equals what?
$1.5.
That's right. And if you look at the 2-point cross, which is further up in the PowerPoint, that's pretty much the number that you get. How we do for time? We're doing okay. Might have time for the second one if we're quick. All right, and then you just do the same for, oh, let's not rub all of those out. Now you want to do for here, right? So between DM and M. So you're going to be looking for ones which are plus plus M. Plus plus M is going to be that one. And the opposite of that will be WDM plus. WDM plus, going to be that one. Always just have to remember that whole reciprocal deal, right? And so that's 100, let's write this up for completeness sake. That's going to be plus plus M and WDM plus, which is 165 plus 160 plus, Levier said, double recombinants times 100 is what? 33. Thank you, Gabriel. And so now what we can do is just work out. Has everybody got this before I rub it off? I'll post the pictures later anyway, but just in case. Oh, okay, good question, Ali. So how you know DM is plus plus M and M is, okay, so that's why, Let's tilt this up a little bit. Could be that you weren't seeing this up there too as well, which is always a struggle. So that's why, so this is our gene order, right? This is the correct gene order, which we get from the double recombinants, right? These ones down here, right? And we know that DM, this one, is the one that moves, right? Because the original parental chromosome is plus, 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 and WMDM, right? And so that the one that moves has to be the one that's in the middle, right? Because it's the result of two recombination events, two crossovers between the three different genes. And so in this case, DM is the one that's moving, right? This one here. So DM has to be in the middle, right? Now, once you have that, now you can just put a cross anywhere. So say like this one is, we're working out recombination frequency between DM and M, and the corresponding crossover is going to be here on the other chromosome. Does that make sense? So once you do that, and this is why writing the correct gene order helps, right? Because otherwise you get all kinds of confused. Then all you have to do is read it off, literally. So one recombinant, single recombinant that is going to be plus, plus, and then the one after the cross on the other chromosome, the X mark, which is going to be M. So that's going to be our plus, plus, M. And then you get the opposite, right? Because it's always going to be a reciprocal. So you'd get a WDM from the original recessive parental chromosome and the plus from the wild type chromosome. That's going to be your WDM plus. Now these ones, for time's sake, I didn't rewrite them in the correct order, right? Because you don't really need to. All you need to look for is, where do I find a plus, plus, and an M? That's plus, plus, M. So the opposite has to be WDM plus, and so I look around, I've got to find a plus and a WDM plus WDM, and that's what I've got, right? So that's why I use these in that calculation. Cool, I'm very glad, Ali. Thanks for asking the question. Okay, I'm going to take another picture of that with the extra squiggles and everything. So you can now order this, and I actually got these data from Flybase to be able to actually do this. Right, so this is an actual real life example. It's just not made up gibberish and words and stuff. Right. We now have WDMM. This is 1.5. This is 33. And from here to here is 34.5 centimorgans. Right, so that is your map. Your linkage map, right? It's obviously not a physical map. We'd have a whole load more numbers on there if it were, but that's your linkage map for those three genes. Unfortunately, I kind of, ran a little longer on time. That's kind of why I wanted to get that other stuff done first. So what I would very strongly suggest you do, all of you, even if this all makes kind of complete sense, is do this one as a practice. Okay, so it's exact same principles. You look for the parents, which have the largest number. You look for the double recombinants, right? So that also gives you the gene type, right? So these are the two parental chromosomes, essentially. Plus, 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 and recessive, recessive, recessive. You then look to the double recombinants and see which one moved, right? So which are the double recombinants here? They're these ones, right? And these are actual fly genes as well. These aren't made up. They do use really irritating names for fly genes, unfortunately. It's one of my pet peeves, along with giant pandas. So, which gene moved in these two? And again, the top line is the recombinant chromosome that you care about. The bottom one's just from the other parent. Which gene moved? So again, parent is plus, 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 or recessive, recessive, recessive. In our double recombinants, we have plus, plus, SS, or STE plus. Which one moved?
The SS.
Correct. I know someone said that, and thank you, Gabriel, you got that one too. Okay, so the gene order has to be what?
The SS in the middle.
Correct. So it's STSSE. All right, now we're out of time, unfortunately. But what I would like you to do is just go through that, and we'll catch up and see if everybody got the same numbers on Tuesday. So this is kind of like your homework. We're not going to go through this like we have with the others because we've got other stuff to talk about, but we'll just catch up and make sure that everybody got the same numbers for this 3-point cross. And I think, Tanya, you probably know more about this than I would, that there are examples to work through in the textbook. Is that the case at the end of the chapter?
I'm not sure. There's always questions like that, but I got like really confused, so I stopped like reading.
Well, maybe go back and look at them, because as you can see, there really isn't any particular magic to it, right? It's really quite straightforward. As long as you follow that kind of order, it helps turn this horrible mess of numbers and letters into something that's really quite easy to figure out. Okay, quick question from Ali. When's the next exam? It is on the 18th for you lot, I believe. Let's just double check. Well, go down a little bit further. Yes, so it is not next Thursday, it's the Thursday after that, so 2 weeks from today. All right, everybody, I think that's pretty much it from me. If you have any questions, oh, you're welcome, Ali. If you have any questions about any of this stuff between now and Tuesday, just shoot me an email and I'll get back to you as soon as I can. All right. Thanks very much, everybody. Have a good weekend.